Hello everyone. So in this video, let us have a short look about the other techniques of thermal processing. So in the last video, we had seen about the basic techniques that are involved for the food preservation. So in this, we can deal about the sterilization and the pasteurization technique that is most adoptedly being preferred in any food processing industry. So coming to the first, pasteurization. So everyone would have known about this very common term, pasteurization, because in day-to-day life, like we are consuming, or each and every individual has been consuming milk. So milk packet is the place where we can find many known words like pasteurized milk, standardized milk, etc. So likewise, this pasteurization is a term that is very much more common among not only food technologists but even to the common people. So coming to the purpose of pasteurization, so why we have to do the pasteurization? So mainly we are going to ultimate aim is to preserve the particular food. Okay, so we have to, in order to preserve, we have to destroy the heat sensitive microbes or the organisms that are present within the food material. So for that, here pasteurization is a technique where any food has to be heated to less than 100 degrees Celsius, so that the enzymes or the sensitive organisms that are present will get destroyed or killed. Okay, so this comes under the category of very mild heat treatment process, since the temperature is less than only 100 degrees Celsius. Okay, so mainly we all know it is being done to preserve the shelf life of the food. Next, coming to the temperature. So as I told, the pasteurization is being most adoptedly done for milk. So milk in milk itself, based on the requirement or the end quality or the end product of the milk, based on that we have different types of pasteurization methods being adopted. Okay, so in this table you can see there is a variation in the time and the temperature. So as I told, mainly with the time temperature combinations only we are going to preserve any particular food material. So starting from mild heat process, that is 63 degrees Celsius, we can go in for pasteurizing temperatures. So by doing this, what happens is that at any temperature there are only like two categories of microorganisms that are being targeted. One is the thermoduric and the thermophilic type of organisms. So thermophilic organisms are nothing but those that will even survive when it is being exposed to very higher temperatures. So those examples are like streptococcus and the lactobacillus. So those types of bacteria also comes under this group. Thermophilic is nothing but they not only survive relatively higher temperatures, but require few temperatures, like very mild temperatures are alone required for their growth. Coming to the methods. So method is nothing but there are actually we can look into the basic method that has been adopted for milk. So in this diagram you can see there is a standardized vat that is being present. Inside a vat there will be a stirrer, and with that help of the temperature thermometer and the thermometer and temperature control, and also an valve will be present outside. After the pasteurization process, the milk will exit from the exit valve. Okay, so in this mainly the holding time that is for milk inside that particular vat will be like only 30 minutes of time, where it will be exposed to nearly 63 degrees Celsius, and it is like most commercially practiced method in any milk processing industry. So the other methods of pasteurizing is nothing but the high temperature short time method. That is, we are going to just expose the milk for 15 seconds for about 72 degrees Celsius, and also HTST method, like it involves a plate heat exchanger. So it is nothing but milk will flow along the series of plate heat exchangers, after which it will be carried on to a holding tube, and the balance tank will be present in order to maintain the temperature of the milk, because as when the pasteurization is over, it has to be chilled down to 4 degrees Celsius. So only a holding tank is being placed in this method of HTST. Then coming to the sterilization. So thank you. Let's continue the sterilization in the next video. Thank you.